हाँ। विद इन टाइम। Good morning, sir. Good morning, respected teachers, colleagues, and juniors. My topic is a complex relationship between PSC and colitis. I have divided my presentation into following subheadings: PSC and IBD phenotype, pathogenesis, where I'll discuss about gut liver axis, immunological and microbiota, effect of PSC on IBD, effect of IBD on PSC, effect of drugs on PSC IBD. So when we look into primary sclerosing cholangitis, it is a cholestatic liver disease which is characterized by chronic inflammation of the intrahepatic and or extrahepatic bile ducts with obliterative fibrosis leading to progressive destruction of the biliary tree. Usually the median time from diagnosis to PSC-related death or liver transplant is 10 and 21 years. It is a more male predominant disease with 60-70% being males, and the age group it is in 40 to 60 years. When we look into the association of PSC and IBD, around 60 to 80% of the patients of PSC have an accompanying IBD. The asymptomatic patients can still have intestinal involvement, which may be underreported. And the prevalence of PSC and UC and Crohn's disease is 3 and around 1.3%. What are the challenges in understanding PSC and IBD? The issues are the subclinical forms of PSC are underdiagnosed even in patients with known IBD. And the blood-based screening, which focus on alkaline phosphatase, can lead to false negative results and underdiagnosis at early stages of PSC. So the screening of bile duct irregularities by MRCP has increased detection of subclinical PSCs by around 15%. And also long subclinical phase of PSC is there, which can be up to 4 to around 35 to 40 years prior to the diagnosis. When we look into genetic risk factors in PSC and IBD, it is basically a multifactorial disease where genetic and environmental factors also play an important role. When the first degree relatives are looked into it, there's a tenfold increase in PSC and also for IBD around 4 to 20-fold increase. The concordance which has been seen in IBD, it is higher in the Crohn's as compared to the ulcerative colitis. There are risk foci which overlap between all this, but the overall genetic association differs between PSC and IBD more as compared to UC and Crohn's disease. The macrophage stimulating and hematopoietically expressed home box have high expression in liver, which could affect the cell proliferation. And specific deletions of hepatocyte growth factor or MTS1 can lead to liver overgrowth, resistance to pro-apoptotic stimuli, and development of HCC. Hematopoietically expressed home box is involved in transcription factor, which is role in the cell cycle regulation, organ development, and cell differentiation. When we look into the genetic correlation between other autoimmune related disease, what we can say is when we look into PSC, there's a higher correlation which is seen with ulcerative colitis and less to Crohn's disease. But the correlation with IBD is high as compared to other forms. There are various signal nucleotide polymorphisms and the genes which have been associated with PSC IBD, what we can see here, and PSC IBD relapse. And multiple gene-wide association analysis have been done, and they have identified various genes which are involved in the genetics of the PSC IBD. I'm not going to go in details of this. So when we look into the pathophysiology of PSC IBD, we have to look into liver and gut. So in this, we need to have animal models to decipher the mechanism and the causality of cellular molecular communication between the liver and gut. The chemical model of sclerosing cholangitis are the mice models which are fed with di-ethoxycarbonyl 1,4-dihydrocolidine. So to assess the colitis severity, a mice model was developed. Here they had also looked into the FOXP regulator cells infiltration by PCR and flow cytometry. And the IL-10 negative mice, which are deficient in IL-10, developed spontaneous colitis, were crossed with MDR2 mice, which mice have an increased chances of fibrosis and chronic liver disease. And these are the model for developing sclerosing cholangitis. What it was looked into it is when they had crossed the mice in the local SPF microbiome, what it was shown in the endoscopy of the IL-10 negative mice was the more chances of colitis as compared to the mouse model which developed the sclerosing cholangitis. When they had looked into the FOXP3 regulation, what they had found was there was a high FOXP3 regulation which was seen in the IL-10 MDR mice. And again, this diagram again shows the higher FOXP3 regulator cells as compared to the IL-10 negative mice which there are spontaneous colitis. So what we can see is that IL-10 negative mice has a higher chances of colitis as compared to the MDR2 crossed over mice, which is a model for sclerosing cholangitis. So there is a probable effect of cholangitis, which reduces the risk of colitis in this mice model. When they had further seen whether the administration of FOXP deficient cells into the mice, what was the effect of the colitis? What they had found out was in RAG1 model, which had FOXP3, and RAG1 MDR2 model, which didn't have FOXP3, the colitis severity was slightly higher in this model, which didn't have FOXP3. And when they again looked into the FOXP3 expression, this was reduced in the FOXP3 deficient model. So FOXP3 is a regulator of inflammation, and increase in FOXP3 is associated with the reduced colitis, while reduction of FOXP3 is associated with the increased risk of colitis in PSC model. So, sclerosing cholangitis, there's no protective effect of colitis if there's absence of FOXP3 cells, and colitis itself can promote the priming and migration of pro-inflammatory immune cells from the intestine to the liver, which can aggravate immune-mediated cholangitis, and it can also have a dual effect. So when we look into the role of immune system and immune cells in the migration, what we know is the recruitment and homing of the immune cells to the liver and the intestine are tightly regulated by the chemokines and the adhesion molecules. There can be a dysregulation in the expressions of the chemokines in the PSC IBD, which can result in the aberrant lymphocyte trafficking, which can contribute to the progression of sclerosing cholangitis and colitis. So the various adhesion models and chemokines which are seen in liver and which have been increased in PSA are like mucosal adhesion cell adhesion molecule, which is usually not expressed in liver, but in PSC liver it is increased. Also vascular adhesion protein, intracellular adhesion molecule, vascular cell adhesion molecule are also increased in the PSC liver as compared to the normal liver. When the chemokines are looked into it, the chemokines such as CXCL12, CXCL20, 21, and 28. Again, there is an increased expression of these chemokines, which are seen in the liver of the patients with the primary sclerosing cholangitis. So the important one in this chemokines is a CC25 ligand, which is specially present in the intestine and the inflamed colon. Basically, it helps in the gut homing of the CCR9 B cells and T cells by activating the alpha-4 beta-7 binding to the mucosal adhesion molecules. And this moiety is upregulated in patients with PSC, which results in the recruitment of T cells activated in the intestine to the liver. So what is the aberrant homing which is seen in the gut and the liver? So what we can say is these are the antigen which are present in the intestinal lumen. So this is the lamina propria. So the dendritic cells act as a receptor for the antigen. So what happens is the dendritic cells, once they accept the antigen, are taken over by the lymph nodes in the mesentery. Here the naive T cells are there, which have CCR7 present and T cell receptor. So in presence of retinol dehydrogenase, retinol is converted to retinoic acid, and the binding of the dendritic cells happens to the naive T cells. And this naive T cells become gut primed, and they get alpha-4 beta-7 along with the CCR9. What happens is this gut prime T cells can again get translocated into the lamina propria, because here there can be an increased expression of chemokines such as CCL28, CCR10. What happens is this gut prime lymphocytes through the venules and to the portal vein, they can get transported to the liver to the portal vein. So what happens is, as I mentioned, that mucosal adhesion cell adhesion molecules, their aberrancy is there. So, and also CCL25. So this gut prime molecule can get transferred into the sinusoids here. And here, due to increased expression of chemokines such as CXL12, CXCR4, and also because of the expression of CCL28, the concentration is high in the peribiliary region. And also this has been also shown in the biopsies where the peribiliary cells have higher gut-primed lymphocytes as compared to the normal patient. So, but what we have to look into it is the homing of the gut-primed T cells. It is not specific to the PSC. Additional factors can contribute for this thing. And the mechanism which controls the recruitment of specific T cell subsets in PSC IBD remains largely unknown. And the migrating lymphocyte, which recognize a common antigen or a bacterial translocation to the liver, can only be speculated. And there is a lack of clarity to which extent the lymphocyte trafficking can occur from liver to the intestine. We only know about from intestine to the liver what the trafficking is there, but we do not know much about the trafficking from liver to the gut. Then we look into it is the bile acid and the bile acid receptors. So what we know is there's a genetic risk factors are shared between the PSC and ulcerative colitis in form of polymorphism in IL-2RA. And this is important for the function of FOXP regulator T cells. There is a dysregulation which is seen between the FOXP T3 cells and the pro-inflammatory cells, basically the interleukin 17. And this has also been shown in multiple autoimmune diseases that they have a role to play, along with PSC and IBD. So when we look into the bile acid, what is this? Is a receptor or a retinoic acid receptor, orphan receptor gamma T. So this is involved in the interleukin 17 helper T cell proliferation. So what happens is bile acid can cause increase in FOXP3 cells and the reduction in ROR gamma T, which can reduce the helper T cells. When we look into the FXR agonists, which are bile acid receptor agonists, they cause reduction in the interleukin 17, interferon gamma, TNF alpha, nuclear factor kappa B, and interleukin 17. So this can get causes an upregulator of the T regulator cells. So whether the individuals who have reduced in number of FOXP3 cells or they have a genetic susception can lead to the development of PSC and IBD if there's a environmental or microbial trigger is a question which we have to ask. Then when we look into the role of bile acid and their metabolites, what we see is the primary bile acids are synthesized in the liver and the metabolism occurs both in the liver and intestine. There's an enterohepatic of circulation of the bile acid and the metabolites occurs, which serve as a key component of the gut and liver axis. So a mouse model was developed to study whether the acute colitis which develops has an effect on the cholestatic liver injury. So they had taken the wild type of mouse model, 10-week-old, and they had taken the MDR2 negative mice. This is a mice for developing fibrosis, and they have fed this mice with a dextran sulfate sodium for 7 days. So DSS causes acute colitis. This has been used as a mouse model, which was reflected in this study by the loss of weight, watery diarrhea. And when they had looked into the effect of liver of acute colitis, what they had found was this is the mouse model, which is MDR2 deficient with DSS, and this is the plain MDR, and this is the wild type of mouse. The AST-ALT reduction was there in the mouse model, which had a sclerosing cholangitis. And also same thing what we can see is for alkaline phosphatase and AST. When the same mouse model, when they had looked into the amount of the serum bile acid complements total bile acid, again, it can be seen that the mouse model, which was just having fibrosis as compared to one which has developed sclerosing cholangitis, the mouse model with sclerosing cholangitis has reduced in number of bile acids. Also, when they had done the further gene set enrichment analysis, what they had found was the CYP7, NTCP, organ transport protein, bile salt excretory protein. Their reduction was seen in the patients with mouse model with the MDR and which were treated with dextran sulfate. So colitis has an effect in fibrosis, but when the colitis severity is more, this can be speculated whether it is associated with a reduced liver injury in sclerosing cholangitis. And suppressions of pathways involved in bile acid secretion, cholesterol metabolism, and xenobiotic metabolism by cytochrome P over 50 were reduced in DSS treated mouse model. When we look into the various bile acid metabolites which are seen, these are like oxalithocholic acid, isoallolithocholic acid. These are the ones which regulate the balance between the helper T cells and the FOXP T regulator cells in the intestinal mucosa. It has been seen that the 3-oxalithocholic acid inhibits the T cell differentiation, while isoallolithocholic acid induces the FOXP T regulator cells. So there's the bile acid metabolites can again alter the intestine immune system. Usually, the both metabolites were absent in the germ-free mice, and they have a critical role in development along with the microbiota for the PSC. When we look into the activation of FXR receptor by bile acid, what we see is bile acid which are absorbed in the terminal ileum, they induce FGF19, which leads to the further reduction of the bile acid synthesis. And upregulation of this receptor can cause a compensating mechanism in response to cholestasis, and FXR has a protective effect on cholestatic liver injury. Now, when we look into the role of microbiota in PSC and IBD, what we have to ask is whether microbiota changes which are occurring in people with PSC and IBD are causal in the development of the disease, or if these changes in the microbiota are the consequences of the disease itself. So, when we look into the microbiota which is seen in PSC and IBD, patients with PSC as compared to healthy controls have a reduced alpha diversity of the fecal microbiota, and there's an increased abundance of more pro-inflammatory bacteria such as Enterococcus and Fusobacterium, and the Enterococcus levels correlate with the alkaline phosphatase levels. And PSC, IBD versus ulcerative colitis alone has an increase of Enterococcus, Streptococcus, Veillonella, which is seen. So our stool microbiota study was done in which the stool samples were analyzed from patients with PSC, and they had been matched body mass index and gender wise, and other than that they have ulcerative colitis and Crohn's patients, and they had done a 16 sRNA DNA sequencing. So what they had found out was when they had looked into the various microbiota such as bacteroids, firmicutes, this is the healthy control, this is the one with the PSC only, this is the one with the PSC and UC, this is the one with Crohn's, and this is UC. So what they had found was the firmicutes reduction was there in the patients with PSC, and along in further reduction was there along with the patient who had an IBD, and there was an increase in the bacteroids. When they had done again the subgroup genus-wide analysis, what they had again found was the Faeculobacterium as compared to healthy control was seen as a reduction in patients with PSC and PSC IBD, and there was an increase in bacteroids which was seen along with roseoburia. So microbiota definitely differs in PSC IBD and patients with plain PSC. Also, various studies have seen the germ-free NOD mice, which had shown less extensive bile duct damage as compared to the conventional gut microbiota. And antibiotic treatment have been shown to increase hepatobiliary damage in the MDR2 mice due to increased bile acid accumulation. And transfer of microbiota from people with PSC into germ-free mice can reduce the intestinal defect. And various bacterial metabolites are there along with anti-glycoprotein 2 and anti-neutrophil cytoplasmic antibody, which can cause the development of severe disease and shorter transplant-free survival. So whether a specific microbial composition or bacterial antigen could serve as a trigger or aggravating factor in genetically susceptible individuals is a question. I'm not going in detail. This is the same thing what I had talked about, the primary bile acid, the microbiota, and the gut hormone, how it causes the aberrated cell trapping, intestinal dysbiosis, and altered bile acid food in patient with the PSC and IBD. So PSC IBD is different from plain PSC because majority of patients with PSC IBD have phenotype which resembles ulcerative colitis. The PSC IBD is distinct because it causes extensive pancolitis, extensive right-sided dominance, frequent rectal sparing, and a backward ileitis. So in patients with PSC IBD, that's why definitely a colonoscopy has to be done to look into the intestinal inflammation. This was again a meta-analysis, I'll not go into detail, where they had seen that as compared to adult and pediatric, there was a higher risk ratio of these patients with PSC and IBD having a pancolitis. And this, they had looked into the rectal sparing. Again, rectal sparing was higher in patients with PSC IBD and also backwashed ileitis. So usually the classical ulcerative colitis, which is seen, there's a decrease in inflammatory activity from distal to the proximal colon. But in patients with IBD, there's a more right-sided predominance which is seen. So when we look into the relationship of gut and liver, whether it is protective or it is a disease promoting, because majority of the patient with PSC without GI symptoms can still have subclinical intestine inflammation. So when the study, they had looked into the intestinal inflammation in patient with PSC without any symptoms of IBD. What they had found was that this is a zero score and this is 1, 2, and 3. Patients with PSC and IBD without any symptoms can still have inflammatory infiltrates, crypt abscesses, architectural differences, and chronic inflammatory cells. So also there's a increased in patients of PSC with IBD in the expression of FOXP3 cells and reduction in the IL-10 cells. This is in the violet dot. So uh, this patients with PSC, what we have to know is this patients can have still intestinal inflammation on the molecular level, even if they are having an absence of inflammation or clinically. And what is the influence of intestinal inflammation, whether it causes worsening after PSC or it causes reduction? So this question, they had looked into the effect of the colectomy in patients with IBD. So what they had seen was whether in patients with ulcerative colitis, when they had matched with underwent colectomy without colectomy, what was the time frame of development of PSC? And they had found out was in patient with ulcerative colitis who had undergone colectomy versus no colectomy, there was no difference in the time to development of PSC. And the overall hazard ratio was not increased in patients who had undergone colectomy in this patient. And what they had speculated was this was usually due to the change over the time in the PSC diagnosis and the UC treatment. These are the various studies which had looked in the effect of colectomy on the course and outcome of LT or death in PSC. Most of the studies have showed no effect of colectomy on the occurrence of the PSC. While the course of PSC after colectomy, some studies have showed a beneficial effect, others have shown no improvement. So the classical ulcerative colitis is not a driver for PSC itself, and PSC IBD is a definitely distinct entity. And whether optimal control of intestinal inflammation can provide protection against liver disease is a question. When the course of PSC is looked after LT, the model for recurrent PSC is a 1-2 study, because more than 1/4 of the patients develop recurrent PSC within 10 years after LT. So here, when they had looked into the colectomy before transplant and also the IBD presence, colectomy before transplant had a lower hazard ratio, while IBD present had a higher hazard ratio for the development of recurrent PSC. But there are multiple confounding factors in these studies due to organ allocation, surgical technique, and it is not recommended to perform a recurrent colectomy to avoid recurrent PSC. When they look into hepatobiliary malignancy post-IBD, cholangiocarcinoma is the most common one, which affects more than 1/3 of the patients. And there is a high lifetime prevalence of cholangiocarcinoma, and hence screening of these patients is required. Um, when the influence of PSC is looked into along with the complications of the IBD and PSC, a question is to be answered whether how PSC defines the intestinal inflammation and complications. So to study the course between PSC and the clinical outcomes of UC, the authors, the liver transplant and non-liver transplant groups were looked into it. What they had found was the patients who had undergone liver transplant had a reduced activity, and also the number of flares were less. And in this same study, what they had found was in the patients with who had underwent liver transplant, there was a less need for colectomy as compared to non-liver transplant. And liver transplant for PSC reduces the risk of need for colectomy. When we look into the course after LT, if we think that the PSC severity has a suppressive effect on the IBD, then we should think that after LT, there'll be increase in IBD activity. But the course of IBD after transplant is variable, and worsening of the IBDs can be seen in up to 60% of the cases. And various studies have been done which have shown that de novo occurrence and course of IBD after LT. Most of the studies have not shown any change in the course, and some proportion of the patient have shown an improvement, while other significant have shown a worsening. So whether disease inherent factors post LT contribute to the course of PSC IBD has to be considered. When immunosuppression is looked at in after LT, tacrolimus and MMF are associated with a worse outcome of colitis as compared to cyclosporine and azathioprine. And one third of the patient with PSC IBD develop intestinal dysplasia. So also intestinal dysplasia and colitis associated carcinoma is increase in patients with the PSC and IBD, and they again have a right-sided predominance for the C of colon. And hence, when we are doing a colonoscopy for this patient, a complete colonoscopy has to be done, and not just the partial. So when role of bile acid, as I have already talked about into it, is role. The bile acids are increased in the proximal colon, and deoxylithocolic acid and lithocholic acid, they cause direct DNA damage, mutation, genomic instability. And loss of FXR receptors and increase in secondary bile acid can cause increase in chronic inflammation and the risk of dysplasia. When they looked into the effect of medicines when we are treating patients with PSC IBD, usually for the mild inflammation, local immunomodulatory agents such as 5-aminosalicylic acid are used, which have shown efficacy against intestinal inflammation and have a modulating effect on the intestinal microbiota, but are associated with a less severe progressive course. They have not been able to show. When they had looked into other agents such as azathioprine, which is suggested to improve the PSC course, they are conflicting results because one of the Swedish registry showed it was beneficial while the French registry didn't show beneficial. So again, this is the Swedish registry which showed the patients who have been treated with thioprines or anti-TNF agents, they do not have an excess risk of biliary tract cancer, and also there is no excessive risk of having a liver transplantation. Other agents such as vedolizumab also have not shown any significant improvement in alkaline phosphatase levels. When we look into the UDCA, which has a dual effect on the immune regulation, and it can have a beneficial effect, which has been seen in the mice models. So when impact of UDCA was assessed in the colorectal cancer or dysplasia in patient with PSC and IBD, and also with the UDCA exposure, there was no increase in the hazard ratio of whether it shows a protective effect on colon or it causes a worsening. When they had looked into the dose also, this was a low dose, this was a high dose, and their dose was not given. Again, not very significant data is there for the course after giving UDCA. So in summarizing, PSC and IBD have a complex course even before transplantation and after liver transplantation, which is seen in PSC because they have more as a UC. And after liver transplantation, the risk of UC and worsening, they are conflicting data. When the IBD is seen before course, before colectomy and after colectomy, again, there are different studies and they have a complex outcome what they have been mentioned. So in summarizing, what we need to understand is a PSC IBD is a entity separate from the classical UC and Crohn's disease with regard to the clinical characteristics, the genetic association, and intestinal microbiota composition. The microbiota composition, which is similar between PSC with or without IBD, which can point whether liver itself is a driver of an intestinal homeostasis. And to understand the PSC IBD pathogenesis, effort should be made to focus on liver-derived drivers of the intestinal inflammation, and also to study the differences in the intestinal mucosal metabolome, microbiome, and inflammation. And clinical research is required to identify patients with PSC IBD in the IBD population because they have a risk of development of PSC and also dysplasia and malignancy. So future research should study for the prospective studies are required to assess the effect of PSC and IBD because most of the studies which I have shown are either retrospective studies and they have very small number of patients. And the role of artificial intelligence in the early detection of PSC in patients with IBD and whether remission of IBD can impact the long-term outcome of PSC and the role of intestinal inflammation and micro. Thank you.
You covered a lot of ground, Jeffrey, and quite exhaustive review. Uh, I think it's still a very difficult disease. You showed a beautiful path of CCR9 and the mesenteric lymph node. I think this was a brilliant work by David Adams. I recollect very well. And the reason why the disease does not subside or the recurrence occurs is the mesenteric lymph node, which has those imprints of the earlier bacterial insults. So the question I have is, you go back to the diagram. Yeah, this one. No, no, this. Yeah, this one. This the next one. Yeah, here you have shown very well that it is a mesenteric lymph node which takes the CCR9, the top right, CCR20, CCR. No, 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 and it goes to the bile duct. And the question I always had is, how is the CCR10 coming into play, and why is the neutrophil causing this injury? So I was wondering, this is about 10, 15 years old concept. So how is the bacterial injury occurring, bacteria causing the injury to the bile duct?
So in this paper, sir, same they have said that because the intestine takes the antigens are taken up by the dendritic cells and they are further crossed over. And also what they had found was in the peribiliary region, there is an increase of CCL28, which is also seen. So that can be a reason which causes an increase in alpha 4 beta 7 primed gut T cells to be concentrated in the peribiliary region along with the other chemokines such as CXCL12, CXCR4.
There was earlier concept that 8 amino acids of the E. coli are translocated to the bile duct, the cholangiocytes. It was called K antigen. It was Kiran Das who discovered that. And the molecular imprint of those amino acids stays there. So it's a challenging thing. And any other comments from people who are working on PSC or so? If not, thank you,
Thanks.
Daniel. We have a second presentation. Yeah, Seema. While Seema is taking the podium, just to say that tomorrow we have one of the brightest minds in the country, Shashi Tharoor, coming. He will be addressing us 12 to 1. And I hope you will enjoy that. Is Pratibha around? Maybe it's too cold for her. So Shashi Tharoor will be with us 12 to maybe 1:30. So you're all encouraged to ask questions, prepare something. He'll be open house after his brief talks, and there will be Davte Jigar tomorrow in the evening. I think we have Brahma Kumari Shreya. All this is for tomorrow, and then day after, 14th, is the foundation day, where we hope to have our Honorable Health Minister and others around 12 o'clock. With that brief, I thought Pratibha would tell, but do share your concepts and be around to have some best of the brilliant minds tomorrow in the noontime. Thank you, Seema.
This is one topic that I have kind of taken earlier for the Department of Hepatology in their classroom, but that was almost half a decade earlier. So I thought there was much more data that has come since then. And so go back and revisit the same area, that is role of genetic testing in adult cholestatic liver disease and cryptogenic cirrhosis, no conflict of interest. And as you know, this slide very well is the various, ATP8B1, ABCB11. This is for FIC1. This is for FIC2, BSEP. This is ABCB4. This encodes MDR3. And then, of course, ABCB1, ABCC1, which are all also there. These are all bile transporters, which help in the bile transport. And ABCB11 and ABCB4 are also helpful in promoting the FXR, which is the chief regulator for bile synthesis. TJP2 is another area that in children especially is was actually reported for the first time in such large numbers from this institute, from a pan-Indian registry that Vikrant had reported in hepatology recently last year. And Myo5B is another area which is largely seen in children, where they have the transport of ABCB11 from here to the canalicular membrane is what is defective. Now, largely these bile transporters, all of them are actually taking the bile from the inner various substrate of the bile from the inner leaflet to the outer leaflet, except for thick one, which is taking the aminophospholipid the opposite way from the outer leaflet to the inner leaflet, maintaining the integrity of the canalicular membrane. Now, I'm going to concentrate on MDR3 because this is largely the one area which is much more commonly affected in the adult cholestatic liver disease, which is actually which is actually responsible for the transfer of phosphatidylcholine from the inner leaflet to the outer leaflet into the canalicular membrane. And if it is not present, then it causes certain problems with the bile becomes much more lethogenic, which we'll see in the subsequent slides. BSEP has also been reported by adult studies, conjugated bile salts. And of course, MDR1 and MRPR2 are largely responsible for the drugs, which we'll see subsequently. So what is the role of ABCB4 in cholestatic liver disease? The pathophysiology is that there will be the ABCB4 MDR3 gene is on the 7th chromosome, and it is responsible for the phosphatidylcholine blockage that I just told you. This decreases the biliary phosphatidylcholine secretion if there is a mutation, and it causes the loss of micelle stability, increases the cholesterol crystallization, and increased lethogenic bile, which causes small bile duct injury and cholestatic liver disease. These are a few of the studies which came in around the 2020, where people were largely talking about the various types of phenotypes with ABC4. And I think the first phenotype that is largely pediatric is PFIC3, hardly seen in the adult age group, and also the last two, that is transient neonatal cholestasis and the parental nutrition associated liver disease, also largely seen during the neonatal period. But for the rest of them, that is LPAC, that is low phospholipid associated cholestasis, for the intrahepatic cholestasis of pregnancy, adult biliary fibrosis and cirrhosis, chronic cholangiopathy, and DILI. These are the areas where the adult hepatologists need to remember a percentage of these cases will always have a genetic predisposition or will have a genetic association, and they need to be picked up. And the reasons why they need to be picked up, we'll be discussing. So a similar study where they've now, for the first time, I think in almost all bile transporters, we used to feel that the heterogeneous group doesn't have any role to play. And so when we discussed the TJP2, we kept the heterogeneous group away. When I have just communicated the paper of BSEP, we have kept the heterogeneous group away. But if it is ABCB4, then a heterogeneous group also has a very important role to play. The heterozygous group has an important. Now, heterozygous is when he's carrying just one mutation on one allele, the other mutation is not present. So in these patients, it's likely that they may have a transaminases elevation, mild chronic cholestasis, ICP, may have LPAC, may have DILI, CIC. CIC is corticosteroid-induced cholestasis. But if you have a severe variety, which is either homozygous or compound homozygous mutations of ABC, which means that the homozygous means that both the same mutation on both the alleles, whereas compound have heterozygous means that you have different mutations on both the alleles. So these patients will usually present with decompensated biliary cirrhosis or PFIC3. I will not go again into the same, conditions which are being mentioned here, except telling you that the age is very early for PFIC3, whereas the rest, it is later. This is a Chinese study where I'm just presenting a part of this is because these 15 children and 8 adults had these variant available. of ABC4 in them. They were usually patients of cirrhosis, cholestasis, drug-induced liver injuries, and mild liver fibrosis. And they had these variants present either on the ATP, that is ABC transporter transmembrane region itself, or in the in-between region, which is a mitochondrial protein. So I just wanted, because Vikrant was on leave and I did not have all the variants that the Pan-Indian, children group has created. What are the various variants that they have? Otherwise, I wanted to create a slide comparing these with the others, and how much can it help the adults from our data is what I wanted to look at. So you do have a lot of variants, and at least 18 of these variants were also novel variants. Just to tell you, like I was talking earlier, that if you have a patient who is having liver fibrosis and cholestasis and he doesn't survive or he needs a transplant, he's more likely to be a compound heterozygous. Rest of them were all heterozygous. This one was the compound heterozygous. And these are also seen in 15 to 20% of the adult hepatology, adult group. And the adult group needs to acknowledge this and do a bit for them, a little more than just identifying these patients. And we'll discuss that subsequently. The classification for the genotype severity of ABCB4 is that if you have a heterozygous missense variant, it is labeled as mild. If you have a heterozygous PTM is protein truncating mutation, or a biallelic missense variant, then you call them as moderate. The severe variety is largely the biallelic PTM, that is protein truncating mutations, or a compound heterozygous, which may have one side missense and one side PTM. So this is largely the classification that they're using nowadays for ABCB4. Now, the most important and common presentation among adults with ABCB4 mutation is the low phospholipid associated cholestasis, which is usually diagnosed if the onset of the gallstones is before 40 years, intrahepatic sludge or microlithiasis is present, the recurrence of symptoms after cholecystectomy, and if there is a family history. So the study has already reported that in LPAC, you have 79 variants already detected, 61 of them missense and 18 truncating sequence variants, 63 may be monoallelic in 156 patients. Now, these 18 truncating sequence variants doesn't mean that they will only have LPAC and go away. They may have associated ICP, they may have associated hepatobiliary disease, and may also go on to have hepatic cirrhosis and cancer. So we must keep these things in mind when we are looking at these patients. LPAC is more frequent in women and is highly associated with ICP, that is intrahepatic cholestasis of pregnancy, and half of the patients harbor a missense or a truncating variant, which we need to keep in mind. But remember that the presentation of these patients with genetic mutation and those without genetic mutation do not differ from each other. So what it means is that we still have some undiscovered genetic mutation that we need to work on if we are looking at this cohort again and again. The more recent criteria to diagnose LPAC is to have at least you either have two major criteria out of these three, or you have one major criteria or two along with the two minor criteria, and then decide about the the diagnosis of LPAC. This is more recent. And a similar study, in the same study, they've also mentioned that some very typical phenotypic features which are associated with ABC4 are in LPAC are more frequent chronic increase in serum GGT. So this is another indication that this could be another set of patients which you need to keep in mind when you are screening for genetic association, that a more chronic increase in serum GGT, increase in the incidence of microscopic intrahepatic stones with bile duct dilatation, common bile duct lithiasis, and higher incidence of primary biliary cancers. These are areas where we need to look for. As far as the imaging of the LPAC is concerned, ultrasounds show intrahepatic echogenic foci or real stones with dorsal acoustic shadow, but a colored Doppler is little more helpful in bringing out all those artifacts looking comet cell-like, tail-like appearance, which are actually small stones and which may not be picked up on normal USG. Unifocal and multifocal spindle-shaped IHBRDs are seen in almost 85% of these patients. Another point is that some of these, intrahepatic stones may not be picked up on ultrasound, although 85% of them are picked up, but some of them may not be picked up, and then they can be picked up on the MRI T2 weighted or MRI 1. T1, yeah. So this is a point of caution here, that the primary intrahepatic lithiasis is often evaluated with MRI only without having a previous ultrasound. They feel that 81% of the of these irregularities are picked up by the ultrasound. So we should do primarily always ultrasound, because alone MRI or MRCP are not recommended, and they feel that both of these things should be done, and especially ultrasound should be done first. In the liver histology, if you look at this, there's marked portal fibrosis and even biliary cirrhosis. But what is important is within the bile duct, you can see the cholesterol precipitation, which is because of the decrease of the phosphatidylcholine. So, and of course, here you can see that there's almost absent immunostaining with MDR3, which is there in the control group, but not here. So these are the various findings that you'll find on an liver histology in these patients. Two or three more, clinical tips, that if you have a patient with low phospholipid associated polylithiasis, and LPAC will mostly happen in non-overweight patients. So if you have non-overweight patients or maybe patients who have developed their biliary symptoms under 18 years of age, and of course patients who have cholangitis and acute pancreatitis associated with the LPAC, they're more likely to be having a genetic association. Whereas those who don't have a genetic association usually have only cholecystitis associated with them. Here, they have also looked up ABCB4 variant, that is 711A-T. 711A to T, they have looked up in 2 areas in Poland in patients with PBC. And what they found was that this is definitely a very important risk factor, along with gender and younger age, if you have patients with PBC. For liver fibrosis in patients with PBC. The same was not seen in patients with PSC. So it's going beyond even PBC and PSCs. We need to be, careful about who are going to develop liver cirrhosis, more likely to have genetic predisposition. Genome-wide association studies for gallstone disease in Ireland picked up a missense SNP and a frame shift insertion, and they said these are very strongly associated with gallstone disease, ICP, liver fibrosis, liver and gallbladder cancer, and cholangiocancer. We need more such studies in other populations. Intrahepatic cholestasis of pregnancy, as we all know, is usually happening in the 2nd and the 3rd trimester. Serum concentration of estrogen and progesterone, it usually reaches the peak around that time, and most common pregnancy-related disease is ICP. It is seen as an incidence of 0.05 to 20%, but it's almost largely seen that in this country, Chile, I think Chile is largely seen in 15 to 20% of the cases. In Europe, the prevalence of ICP is around 2%, and 15% of them are attributable to ABCP4. ICP associated with ABP4 will have high serum GGT levels. Whereas ICP associated with ABCB11 and ATPB1 and farsinoid X receptor will be having low GGT. So a few more, clinical tips that you can have to decide about the screening. This is another scientific report of ICP where they have looked up 1954 AG. This is a missense variant in all the patients of ICP as well as in controls. And it seemed to be present in all of them. But when you look at the patients who actually got ICP, they were the ones who had both 1954 as well as other variants. These are the other synonym variants which were also looked at, and only 1954 was not responsible for that ICP. If they had another variant, only then they would have they would present clinically. And more than that, if the patients had just one variant, then their numbers, clinical numbers, or the liver numbers were not that bad. But their liver numbers were much worse if they had at least 2 or 3 variants associated with the 1954. This is the structure for the MDR3 or ABCB4, and this 1954 is supposedly present in the linker area. So it doesn't cause the disease itself, but it is causing the disease only when it is superimposed with the other variants. Another single center study report is the whole genome sequences done in 20 unrelated cases and of molecular spectrum variants, and shortlisted 5 unique heterozygous variants. And ABCB4 was seen in 3 of them, and one of them was actually having a stop gain variant, or what I was just talking about, a PTM, a protein truncating terminating variant here. And this is the patient. Yeah, sorry. So it's important to not just look at the various genes. It is also important to look at the type of the variant that the patient is having. He's likely to be having future problems if he has a more severe variant present. ICP may unmask, and so I say that ICP may unmask an underlying defect associated with mutations in ABCP4, which may predispose the individuals to hepatobiliary disease, cirrhosis, and even malignancies. So a regular follow-up and a genetic counseling of the whole family is very much required, not just screening that one particular patient. And this should be discussed in detail with women for ICP due to ABCB4 deficiency and other genetic factors. This is unrelated to this, but our own group of BSEP, 20% of the mothers were found to be having ICP. Of course, due to logistic constraints, we are not able to give them the complete genetic workup, but this is what is discussed with them. So various studies on ICP and ABCB4 has been thoroughly looked up in all populations. You can see here. So there has been enough data on ABCB4. There is now data also for ABCB11, ABCC2, ATP8P1, which is STIC1, and of course FXR gene, which is NR1H4. Lastly, I will talk about the drug-induced liver injury, which is regularly seen in patients with ABCB4 deficiency, especially with drugs like oral contraceptives, psychotropic drugs, selected chemotherapy drugs, statins, and antibiotics. This presentation, graphical presentation, is for a mother who was a donor to her child with PFIC, but after she did the donation, a year or so later, she developed drug-induced liver injury because she was carrying an ABCB4 mutation. And so in patients with ABCB11 and ABCB4, there's a 3-fold increase in the cholestatic DILI. So if you have a patient you have screened, you have to tell him that there are drugs that he has to keep away, or at least be cautious of those drugs, because he's likely to develop DILI as well. Sirolimus, cyclosporin, verapamil, and vinblastine is something. These are the drugs which are more likely to cause DILI in these patients. And I was always wondering about the patients who have received their mother or father's liver in our cases of ABCB4. Are we keeping away from sirolimus? Are we keeping this in mind all the time? Again, we know that MDR1 and MRP2, MDR1 and MRP2 are ATP-dependent transporters, are encoded by genes like ABCB1 and ABCC2, and we've seen that ABCB1 has been associated with DILI due to decreased expression. So these are further genes which have to be looked into if you're thinking about DILI. This is the largest study done in Europe, where they had looked up all cholestatic liver disease, and they had done 651 sequencing of analysis of patients, of which 184 was FIC1, 252 for VSEP, and 215 for MDR3. And they found from 9.8% in FIC1 to 34.9% in VSEP, and 20% MDR3 were found to be carrying at least one mutation, VSEP disease-causing mutation, and at least 20% of these patients in the various groups received liver transplant. So it is that important. And of course, SNBs were also present in these patients in very high numbers, 35%, 64%, and 72%. Common ABCB4 and ABCB11 genotypes are associated with. Now I'm just giving you 2-3 slides of idiopathic cholestatic liver disease or idiopathic, idiopathic cirrhosis. And here they have found that these patients, this is actually coming from two hospitals in Europe where they found that if you look at ABCB4, two of these mutations for ABC4 and ABCB11, two of these mutations in patients with cirrhosis vis-a-vis those of controls, then at least this mutation and this mutation turned out to be having a significant association with cholestasis. Cryptogenic cholestasis in young adults, all these mutations are required to be done. All these genes need to be looked into. And they do find various, ATP1 had just one variant. This had ABCB11 had two variants. MDR3 had two variants, and TGP2 for the first time had three variants present in these patients with cryptogenic cirrhosis. This is a study done in India itself in SGPGI and in TMH, Tata Memorial Hospital, where they did a genome-wide study for several markers in the chromosomal region of 7, harboring both ABCB1 and ABCB4 genes, and they found that there were some notable SNPs identified here, which had a good odds ratio of 1.47 association with the malignancies. And they found that the sibling relative risk was as much as 3.15 if the patients were carrying these SNPs for a gallbladder cancer. I'm just quickly going to go because whole exome sequencing has been found to be very effective in children. Is it effective in adults or not needs to be seen. Whole exome sequencing means that we are only sequencing the disease causing protein. That is all that we are. So it becomes much less expensive than the genome whole genome sequencing, and it has less volume and easier to report also. But another proposition that I have here, just like we have a transplant board and a tumor board, we should have a genomic round or a genetic board as well, where we should have some genetics people from inside and outside, and maybe clinical groups which are working within and outside should also participate in that genomic round. Because without the genomic round, we are just, kind of indulging in the open air with some of the yield is not going to be enough for us, and yield is never going to be cost effective. So we should have this. So if you see these patients in this one study, they had tried to look up cryptogenic cirrhosis, non-obese, what is adult hepatology always talking about, lean NASH. So they use lean NASH also, idiopathic cholestasis, help patients, idiopathic cirrhotic portal hypertension. And what they found was that they have at least 5 or 6 of their patients, out of those 19 patients, 5 or 6 of their patients had a diagnosis by the end of this genomic round and by the end of the screening. So what we need is a genomic board or a genetic board. Something has to be on those lines. We know that PFIC genes are much more likely to be found than others when we screen, as is seen in this one study, where they have seen that the gray color means not much variants, but blue means at least one variant, and the red means more than 2 variants. So this is how the things are at this point. It's largely the PFIC genes which are largely being seen, and they're largely being, picked up. 53 Turkish adult patients, idiopathic liver disease, they went through this whole exome sequencing. You look at the number of patients with genetic diseases and the number of total patients, you do not find much difference between them. So clinically, you may not have been able to decide which one is going to be genetic disorder. But yes, cirrhosis was more commonly seen in these patients. But at the end of it, they came up with at least 10 patients with various diagnoses could be looked up. If you look at these genes, they're not largely PFIC genes. There are other genes also here, and they need to be now. FOCAD is one gene that we yet to see even in pediatrics here. There are very few FOCAD patients. It's an addition molecule or addition protein, the gene for addition protein, which is there, and it has recently been reported in children also, but it is now being reported in adults as well, has been seen in adults, in those with cryptogenic cirrhosis. So a clinical exome sequencing, again, giving you a lot of, at least a diagnosis, giving a diagnosis to a cryptogenic cirrhosis is what you can see here. Various patients are getting a diagnosis. And hence I propose that we should have a protocol where we or any patient with increased enzymes or bilirubin should look at the bile ducts. If they are not, if they're dilated, they're not dilated, then more so you should go ahead and look for the PBC if it is not there. But in my case, I will say now, even if it is dilated, even then go forward and ignore, if PBC is not present, then look at the GGT. And if the GGT is slightly increased or normal, you look up the bile acids. And if the bile acids are increased, then ABCP11 deficiency is likely and PFIC1 is likely. But if there is normal bile acid concentration, then in that case, a bile acid synthetic defect is something that you should look for. And ABCP4 if its GGT is high. Our own results from the this thing for MDR3, 120 or so patients, half of them are being ours. We have very few mild, we have largely severe and moderate variety of the genotype categorization. As I told you earlier, they're largely coming in young age. And of course, when we do the multivariate analysis, logistic regression analysis, we find that genotype severe versus all other genotypes seems to be having a adjusted OR of 5.65 to predict poor outcome in these patients. So my take-home message is that ABCB4 variants are common cause of genetic intrahepatic cholestasis in young adults, but the new genes to look for is ATP18, B8, B7, and TJP2. Especially in areas of LPAC, ICP, DILI, cryptogenic cirrhosis, elevated transaminases, and persistent high GGT and high bile acids. If detected, then these patients should not be just labeled as ABCB4 or any other mutation that you pick up, but they should be counseled and there should be a very detailed family counseling. And we should keep in mind that these are patients, some of them may become even hepatic cirrhosis. Thank you. And here I think that it's time we talked about the adult registry, and the adult registry of, for PFIC should join the Indian PFIC registry that we're already having, maybe at the level of APASL or at the level of ILBS itself. But the man who's doing this, all the legwork and the hard work is Vikrant Biharilal. Thank you.
Thank you. Seema, can you go to the previous slide? I think it's a very key slide and I want all residents and faculty to pay attention. So if you have a cryptogenic cirrhosis, which you sometimes put it as NASH or MAFLD or FASLD, it's not correct. I think what she has shown very clearly that cholestasis genes like ABCB4, ATP1B8 or 11, all these are cholestasis genes. So when we don't understand, I think we should look into cholestasis gene. Maybe we can add the telomerase-based genes also. So if you have a patient who has young age cirrhosis, high gamma GT, and you can't explain, I think we should go into
I suggest, sir, we should have a genomic.
I totally endorse. I was going to come. We should have a genomic board. We should learn what we are not aware of and don't accept our ignorance. So thank you, Seema, for bringing it to adult hepatology forum. Thank you. Pratibha, we were waiting.
Yes,
Quick
sir.
announcements.
So get ready for turning sweet 16. 2 days of extravaganza here. So tomorrow we'll start at 11. The Dawati Jigar participants to be ready at the stalls at 11. And 11:30 in the auditorium, the Tambola and the fun games will start. So Tambola.